good evening to all of you. Hello Manpreet, how are you? Gayatri, good evening. Jinal, good evening. So is my voice is audible to all of you? Yes, we have Sakshi, we have Ankita. So a very good evening to all of you. How are you all? Yes, Manpreet, I am also good. I am good today. Because of some technical errors yesterday, I am unable to take. That's why I am little bit early today, so that we will have not any type of problem regarding the connection or connectivity issue. So yes, Ankita, how are you? So how are you all? Kindly let me know. And is my voice is audible to you? Yes, yes, please join. So what we have done yesterday? Yes, and Ankita, I am good. Jagriti. Sanjukta, yes, good evening to all of you. So how are you all? Everyone is fine, okay? How is your day today? Okay, thank you Manpreet, thank you for the voice. Thank you. So how is your day today? What you have done today? Everything is fine? Yes, yes. Yes, Simran, good evening. How are you? And how is your day today? Everything is fine? How are you all? Just waiting for the students to join because it's we are 50 minutes before, but it's okay. Just yes, Ankita, good, very good. So just waiting for the other students to join, then we will start our today's lecture. And one thing I want to tell you that today is a very interesting topic. We will discuss about some type of hormones like steroid hormones. So you all will be fine today. Yes, please, good evening to you. So is everything is fine? Yes, we have Muktik Tiwari. So good evening, a very good evening to you. Mitali is also here. Mitali, good evening. Shivangi, how are you, Shivangi? So lots of students are there, the good students. So a very welcome to all of you. And I just apologize for the yesterday technical error. Because of it, I am unable to take your yesterday lecture. Yes, Shivangi, I am very much fine, thank you. And how is your day today? I think today is a working day. Today is Wednesday. Maybe some of you have some type of college classes. Maybe these type of activities may be there because today is the working day. So kindly let me know whether you have online classes or there's exams. What are you doing these days? Yes, Nitya, good evening. How are you, Nitya? So I'm asking that what is your routine these days? Is there regular online classes from your college? Yes. That's what I'm asking, Shivangi, that you have assignments are there, then of course you have some type of theory papers or practical papers. So it's a very good effort of all of you that you are here even after so much of busy schedule. So that's great. That's totally great. Yes, 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 yes. That's what I'm saying. That you have regular online classes, the theory classes, but how you are able to conduct online practicals? It means the teacher is there in the department who will demonstrate you everything regarding the practical. That means theory is okay, that we will discuss the things theoretically, but how practicals are possible? Can you let me know? Yes, Nitya, I am very much fine, thank you. Yes, Shraddha, I am very much good. Okay, Shivani is saying that they demonstrate and shows video. Okay, very good. And you have to make practical files also, in which you have to draw the diagrams with the help of pencil. This type of work also you have to run. Yes, Prachi, good evening. Ankita is saying virtual labs. Okay, very good, very good. Yeah, virtual labs is a very new concept. The whole concept will be clear to you by those videos basically. So it's also a good technique. It's a good one. Sanjukta is saying she is currently pursuing my master's. Okay. Oh. Actually, Sanjukta is very busy up to 6 p.m. She has tests, assignments, practicals. Oh. Yes, that is also one problem. Then everything is online. So the teachers think that students are free. So they put lots of work only on the students. You have to do this, you have to do this, you have to make assignments and everything. This is also one problem. And of course we will miss those things which we have done in the colleges, like when we are basically there in the college, like when we are not in a mood to take a lecture, then we go to a bunk, or to go to the college canteen. So these things are basically very important in students' life, to go to canteen and bunk the lecture, or these type of gossips are always there in the colleges. So this is the one thing which students must miss. Okay. Okay, Shivangi saying word and written both. Okay, okay. And they have to make practical files also. Oh my god. And mail it to the teachers. Yes, ma'am, we have to make lab reports via virtual labs. Okay, in which our professors record videos at the lab and show it to us. Okay, okay. Yes, Shivangi, I agree with you, because when the online just start in the month of May or June, July, so we are very happy. But now it's like that when we will go to college or we will meet our students, this type of online interaction is okay, but the face-to-face interaction or the personal interaction is also very much important in our lives. We are now on the phone always, either on the WhatsApp, or I think the whole life is oriented towards the phone only. So that is the one thing which I don't recommend to any of you. Sindhukta is saying she wants to bunk offline classes. Yes, probably everything will be fine soon, and you will definitely enjoy your college period or your bachelor and master's period, because this period will not come again. When you were in your master's and in your bachelor, the life is very different. But after it, even in PhD, the life is different. But when you are in like in my position, then you have, the life is totally changed. So I request all of you to kindly enjoy your bachelor or master's phase also. You will miss these things later on. So just be with your friends, be with your family, and enjoy this phase. As last year. Yes, Mystic is very true. Yes. Oh, Shivangi has also a busy schedule. she has the gate classes from 5:30 to 7:30. That means just from the 20 minutes she may be free, otherwise she has the classes. So yes, you are not gaining any lab experience. Plus, in online classes, what happens? Teacher puts lots of syllabus on the, or you can say lots of things on the students that you can do it, and the students also they think like that we will do it later because we have to read from the videos only. We have to get it from the videos only, so we will take it later also. So this is basically the problem is because the teacher is giving the assignments and then we are there that we will do it later because no one is there who is watching us. Like teacher is if the teacher is there in class, then we have to do work. But if the teacher is not there, how we will do work? So these are the things which are basically, the lack of this online system. I think so. Yes, okay. Shivangi has classes from early 7:30 to 11. Okay. So she has a very busy schedule. Ankita, yes, my schedule is also very busy. From 9 to 5 p.m. I am in my university because teacher has to be there in the university. Or then after 5 p.m. I reach my home. Then from 5:30 onwards I have one online classes and PPT class. Then I have your class. So my schedule is also very busy. But we have to manage it. Oh my God, Shivangi is very much busy. Shivangi, in which class basically you are? You are pursuing masters or you are in bachelor's? Yes, Sanjukta, thank you a lot. Yes, we celebrate Navratris, we celebrate this great, the festival of Mata. We celebrate it, and we also have small girls known as Kanjika in our home on the last day, and even in our family the girls or even the mothers also they take the fast. I think this is the thing in our side. This is the thing we have to do the Ashtami Puja basically on the last day, in which we small girls are there when we feeding them the food. So yes, we are also celebrating it. Yes, Meera is joining now. She is saying good evening. Good evening, Meera. How are you? Ankita saying she is also attending the classes. Okay. And Shivangi is in BSc Honors Biotechnology or in the 4th year. So she has lots of work. I understand your situation, Shivangi. It's just a difficult phase. Even we are on the laptop, or you can say on the mobiles continuously for the study purposes, but we are missing our physical interactions with, basically I am also missing my physical interactions with my students. Like there are lots of students in our side which are from Jammu Kashmir area, or the area which are very in the northeast side, like from, you can say, from Manipur or from northeast side. So sometimes they are not able to get the good connection also. So this is also a problem which we are facing. Yes, Cloris is here. Good evening, Cloris. How are you? Basically just we are waiting for few more students to join. Then we will start the session. I am just waiting that they will join also. Then we will start. So I am just interacting with all of you. Yes, actually I want this type of platform or this online software in which I am also able to see your faces, to see you. Otherwise, how I am very, I am just only know you by your names only. So that is the one thing which I miss in the online platform. If you are there with me, like on some platform, we are able to see each other. It will be much beneficial. Yes. Ankita, I am taking the classes from 5:30 onwards. Okay, Cloris is good. Okay, good. So I request you, if your friends are there, kindly share the link with all of them so that they can join, and we will start our session. Vishali has a online lectures from 8 a.m. in the morning to 6 p.m. and no lunch break or nothing like that. So I don't know how you manage, because it's very difficult for all of you to be on the mobile or the laptop continuously. Even if we are in our home, we can watch any movie of 3 hours only. Then we just want to switch off the phone or the laptop. So from 8 a.m. to 6 p.m. it's very difficult for you, Vishali. Yes, how are you managing? Okay, she is preparing for MPSC exams. Okay. Yes. So that is the thing which I think by online the life is little bit fast. We have to do this, and we are not enjoying the pace of a life. Maybe I'm wrong, I don't know, but this is my point of view. Yes, Arundhati is here. Good evening, Arundhati. How are you? Nupur, how are you? So today, basically, yes, sir. Shall I'm just going on that thing basically now. Yes, Komal, good evening. Nupur is good, very good. Arundhati, how are you? Arundhati? Okay, so today we have to cover. First, let me tell you what we have to do in our lecture series. Okay, Arundhati is good. I am too fine. Thank you. I am fine, dear. So in the today's lecture, we have to do two things. First, we have to complete the one topic which is left in the first day, that is central dogma. It's a very small topic. We will cover it first because some students are there who said, ma'am, kindly complete whole syllabus or everything which is there in the syllabus. So I will complete the small topic which is central dogma. Then after it we have the day 2 lecture, which is basically the hormones. So it's very interesting topic. The hormones, it includes protein hormones, steroid hormones, and we have thyroid hormones. So we will do with the thyroid then after it. And then we have your review paper discussions which you are asking, and even lots of students also message me. I tried my best to reply you and give you the proper way to write it, but sometimes what happens, I am unable to answer or something like that. So I request all of you to just write here, because now I am in front of you, so I will try my best to convey you the things which I want to convey. Okay, so shall we start now? First we start with the topic. We will just cover a little bit of topic. Then in between we will take the break, and in this break we will do this review paper questioning. Okay, so I hope you all are ready. We will start with our lecture series first. Okay, are you ready? Shall we start now? Kindly let me know. So my voice is loud now. It's not low. I will came back in between the PPT to see whether you are getting it or not getting it. Okay. So okay, Jagriti. Okay. Good. So I will come in between the PPT to check whether you are getting it or not getting it. Okay. So my voice is loud enough. Everything is okay. Shall I start now? My voice is okay. I think it's okay, but kindly let me know. Okay, good. Thank you, Arundhati. So let's start with our the remaining topic of the first day, which is central dogma. Let me share you the PPT. So we have to start with the small topic which is left on the previous day or on the last day of the lectures. We have to do the central dogma. We have basically what we have done on the last lecture. We have done the structure of, let me revise fast. So okay. So we have basically what we have done on the last day. We have done what is nucleic acids and the history of it. Then we have done the introduction, their history, the features of nucleotides. What are the nucleotides? What is nucleosides? What are pyrimidines? What are purines? So we have done all these things. So we have done the introduction of this. The history is also we have done. And then the components of nucleotides, we have two types of sugar. You have to you have to keep it in mind that we have a ribose sugar which consists of OH group, and then we have a 2-deoxyribose sugar in which the OH group is absent. So it lacks 2' OH group. Then we have types of bases. We have purines. The purine consists of adenine and guanine. Then we have pyrimidines. The pyrimidines consist of cytosine, uracil and thiamine. So how many purines we have? We have 2 purines. The purines are adenine and guanine, and these are generally the double ring structure. So you have to remember adenine and guanine are double ring structure. Then we have pyrimidines. We have cytosine, uracil and thiamine. If it is a DNA, then it's thiamine. If it's a RNA, in case of RNA, it's a uracil. So we have 3 pyrimidines which are generally single ring structure. So we have 2 purines, 3 pyrimidines. Okay. So these are the structures. These are double ring structures. In case of pyrimidines, we have single ring structures. So these are the nucleosides which are generally made from sugars and nitrogenous bases. But when the phosphate group is attached to it, it becomes nucleotides by the addition of the phosphate group. So we have done this. Then this is the thing which we have to discuss today. That's the central dogma. Central dogma is basically what it is. Central dogma is from DNA there is the formation of mRNA, or you can say messenger RNA, and this process is known as transcription from DNA to RNA. Then from RNA we have to make protein, and this process is known as translation. So from genome to mRNA it's known as transcriptome. So transcriptome is also studied in a detail also, and even we have a lecture series on transcriptome also. Similarly from mRNA we have proteins, and it's known as proteomics, or you can say proteome, and the lecture series is also there, basically focused on these protein molecules. So these are studied in very detail also. So these are nucleosides which consist of sugar and base, and then what are nucleotides which consist of sugar, base, plus we have a phosphate group which is attached to it. And then how the nucleic acid, basically how the DNA is formed. So it is formed by the combining of these nucleotides. So basically what is DNA? It is a polynucleotide. This is one nucleotide, sugar, base and phosphate. This is one nucleotide. So by the combination of many nucleotides we form the nucleic acid, also known as DNA. So this is what nucleic acid is, basically what it is, a polynucleotide in which we have many copies or many numbers of nucleotides. So that's why it's known as polynucleotide. Then we have again the structures of pyrimidines and purines. We have nucleosides. We have done it already. Then the sugar is generally pentose sugar. What do you mean by pentose sugar? That is 5 carbon ring. So we have a pentose sugar. In case of ribose, that is your RNA, we have OH group, but in case of 2-deoxyribose, OH group is missing and it's known as 2-deoxyribose. Then we also done the how the two polynucleotide chains attach with each other. They bind with each other with the help of hydrogen bonds. So we have a connection between the, you can say, purines and pyrimidines. So adenine combines with thiamine. Just keep in mind, adenine combines with thiamine, and then cytosine or guanine combines with cytosine. In case of adenine and thymine, we have double bond. In case of guanine and cytosine, we have triple bond, and these bonds are basically the hydrogen bonds. This is a thing which we have done the previous day. Then this is a combination they are showing that how these nucleus bases. So the two polynucleotide chains generally bind with the help of these nucleotide bases. So these, you can say, nitrogenous bases play important role in the formation of DNA or in the structure of DNA. Then this is the RNA structure. In RNA structure, instead of thiamine, please keep it in mind, instead of thiamine we have uracil. So instead of thiamine we have uracil in case of RNA. So this is the another difference between DNA and RNA. DNA is double stranded, RNA is single stranded. First point. Then another thing is DNA consists thiamine as a nitrogenous base and RNA consists of uracil as a nitrogenous base. So this is the you can say prominent difference between RNA and DNA structures or RNA and DNA. Then these are the phosphodiester bonds which are formed between the polynucleotide chains. These are the pentose sugar. I am just revising it, so I am just moving little bit fast. So we have, what's the full form of DNA? It's deoxyribonucleic acid. Deoxyribonucleic acid. That means the ribose sugar lacks the OH group. The ribose sugar lacks the OH group in case of 2-deoxy or in case of deoxyribonucleotide or DNA. Then we have ribonucleic acid, we have duplex. So we have done all these things before. These are nucleosides. This is a phosphate group which attached. So we have pentose sugar here, we have nitrogen bases here, and we have phosphate group. So these nitrogen bases are basically responsible for the formation of the, you can say, double helical structure or double structure, because two poly chains are joined with the help of the bonds, the hydrogen bonds which are present between the nitrogenous bases. So these are the polynucleotides. Then we have the pyrimidines, which is single ring, purines are double ring. We have discussed it last time also. Then we have these nucleotides which consist of purines and pyrimidines. So pyrimidines, you have to remember one thing, that pyrimidines are single ring structure. Basically they are heterocyclic rings, but the purines are, you can say, little bit more complicated. They have double ring structure, but pyrimidines are generally simple in structure. So this is the thing we have covered the last day also. So I also want to share you one property, that the properties of purine bases. What do you mean by purine? That is adenine. And another one, that they are springly soluble in water and they absorb the in UV region at 260 nanometer. They are capable of forming the hydrogen bond. That is why adenine is forming the hydrogen bond with thiamine, and guanine is forming the 3 bond or triple bond with cytosine. So adenine and guanine are, you know, capable of making the hydrogen bond. So this is the property of the purine bases. Similarly, pyrimidines are soluble at the body pH. They also absorb UV light at 260 nanometers. So both are absorbing the UV light at 260 nanometers. So you have to remind it in mind. You have to keep it in mind. So they are also capable of forming the hydrogen bond. So this is the properties of the pyrimidine bases. So this is the pyrimidine basis, which is a single ring structure. You have to keep it. Then the first example is cytosine. Then we have thiamine, we have uracil. Uracil is only found in RNA. Please keep it in mind. The uracil is only found in RNA. Similarly, the thiamine, if you are that's thiamine, it's only found in DNA. So thiamine is found in DNA and uracil is found in RNA. Then we have purine bases, in which we have adenine, which is a double ring structure. Then we have guanine. So that's all for the. These cycles we will discuss in detail on the metabolism processes. That is why I am not discussing it here. These cycles, like this glutamate dehydrogenase, we will discuss it in the cycles. We will discuss in the another lecture series. So these are all the main points about nucleic acids. Then we have a Chargaff's rule. I told you on that day also, we have a Chargaff rule in which adenine binds with thiamine. That means adenine is a purine and thiamine is a pyrimidine with the 2 double bonds, and guanine binds with the cytosine with a triple bond. So Chargaff rule lie in the structural basis for the Chargaff rules lie in the hydrogen bonds between the bases. G always hydrogen bonds with the C, and A always form the base pair with the T. So according to Chargaff rule, A always form the bond with the T. So you have to remember the Chargaff rule. And even they have shown that GC content, that is guanine and cytosine content, is generally varies from 25% to 75% in different species of bacteria. But the main point is here that A forms the bond with T and guanine with the cytosine. So you have to remember the Chargaff rule also. Then we have the things which we have to do today is RNA basically. So this is the deoxyribonucleic acid. We have already discussed it also. I will not. So this is from how, this is the cell. In the cell we have a nucleus. In the nucleus we have a chromosomes, and on the chromosomes we have the segments of a DNA. So segments of DNA are present on the genes. Genes are present on the chromosomes, and chromosomes are present inside the nucleus. So DNA cannot leave the nucleus. If someone asks you where the DNA is present in your body, so you have to remember, you have to keep it in mind that DNA is present in the nucleus of the cell, and DNA is double-stranded structure. So we have done it already. Then we have this tertiary structure or quaternary structure we have done. Now we will move forward to, so they are again showing the, you can see the hydrogen bondings between the base pairs. Like here, between thiamine and adenine we have a double bond, and guanine and cytosine we have a triple bond. So you know it. Yes, then we have another very important term which is known as replication. So what do you mean by replication? Basically, it is also suggested by Watson and Crick, but later on lots of experiments has been done by other workers. So according to the replication, what happens is, it's a process by which DNA copied itself. So what happened here, if you are able to see, the DNA is double-stranded here, right? So DNA is double-stranded here. With the process of replication, this double-stranded DNA, this one, it will break. So it will become single-stranded, this blue one. Okay, I just make it like, so it will become single-stranded, and then there is a formation of daughter strands from the other side, and we are able to replicate, or we can say copy the original DNA. So basically what is replication? It is a process by which DNA is copied itself, or the number of DNA gets double. That is, it is 1 in the beginning, then it becomes 2, 2 to 4, 4 to 6, 2 to 4, 4 to 8, 8 to 16. By this the number becomes increased. So DNA replication is basically what? It is the doubling of DNA, or it is the copying of the original one. Okay, I hope you know it. So if you are able to see here, this strand, this double-stranded strand becomes separated here. So we have some type of enzymatic reactions, or some type of enzymes are there which will able to break these bonds, and then what will happen? The new strand will form. So from the one DNA we are able to form the 2 DNA. So this is the replication of DNA, and it's a very long process. It's not that simple. It has many steps, but now it comes here, that's what I described. So replication is the replication or doubling of the DNA. Now what's the transcription? Transcription is basically that the formation of mRNA from the DNA, and what translation? Formation of proteins from the mRNA. So this is the three terms you have to keep in mind. You have to know replication, transcription and translation. So this is all about DNA. We have done it previously also, but I just revise it once again. Now we have to know about RNA, because RNA too we have to make the central dogma. So basically we all know that RNA is a single stranded structure and it consists of adenine, guanine, cytosine and uracil. So adenine pairs adenine with uracil and cytosine with guanine. So we have the uracil in case of thiamine. Then we have some types of RNA. Just remind it, we have some types of RNA. We have messenger RNA, we have transfer RNA and we have ribosomal RNA. I think you must have covered it before also. These are the three prominent RNAs, but instead of it we also have small nuclear RNA, micro RNA, then the small interfering RNA. So these are the types of RNA. So let me tell you the functions of these RNA, just functioning of these three RNAs only. So mRNA is basically the messenger RNA. mRNA is the messenger RNA and it acts to carry genetic sequence information between the DNA and ribosome. So what messenger RNA basically do, it takes the information from the DNA and then pass to the another one, or pass the information. So because DNA is the only one which carries the genetic information, so how this information will passed on? With the help of these mRNAs. So we have tRNAs which serves as a carrier molecule for the amino acids to be utilized for protein synthesis. Okay, so tRNA is basically for the protein synthesis. Kindly keep it in mind. Messenger RNA will carry the information from the DNA. First point. tRNA is basically used or basically utilized in the formation of protein synthesis or in the formation of proteins. Then we have another RNA which is rRNA, is a major component of ribosome and catalyzes the peptide bond formation. So what rRNA do? It helps in the peptide bond formation. Okay. So these are the types, these are the mRNA which carries the information from DNA to the other things. Messenger. What do you mean by messenger? Which will carry the messages, which will share the information. So this is the ribosomal RNA. If you are able to see that it is the major part of the ribosomes, which is where proteins are made. Then we have transfer RNA which helps in protein synthesis. So this is a structure of T-shaped structure of, you can say, our transfer RNA. Then we have the central dogma. Central dogma is same from DNA to RNA and RNA to protein. That is, we have transcription here and we have translation here. So process where genetic code is read and transferred to mRNA, and from mRNA we will form the proteins, is known as translation. So the process of transcription and translation is included in central dogma. So if someone asks you what is central dogma, it is a formation of proteins basically from the DNA with the help of transcription as well as translation. Okay, it's a formation of protein synthesis from the DNA but with the help of transcription as well as translation. Okay. So this is basically all which we have to discuss in this section. I will not carry it much further. Then we have proteins also, but this we will discuss later on, because we have to know only about the structure of DNA, but till we are discussing something about RNA, and along with it we also cover something about the central dogma. So I hope you all will get it definitely. Now may I ask you any question also? May I ask you questions? Yes. So So may I ask you some questions? So you all are present, you are listening to me. So kindly tell me, what are the functions of three different RNAs? What's the function of mRNA, tRNA and rRNA? So kindly let me know. What's the function of our mRNA, tRNA and rRNA? Yes, very good, Gunjan. So messenger mRNA is basically the messenger RNA which will carry the information. But let me check what you will ask. So yes, mRNA codes for proteins. No, I am saying what's the function of mRNA. I am not saying what they code. What's the function of mRNA? We have discussed it just now. What's the function of these mRNA? Yes, Komal is also right that mRNA acts as a template for translation. But basically, what mRNA implicates? Why it's known as messenger RNA? Why? Yes, mRNA is used for protein synthesis. Very good. That is, from mRNA we will form the protein by the process of translation. But why it is known as mRNA? Why? Messenger, why it's known as messenger? Yes, Komal. Because the messenger RNA carries the message from the DNA which has whole hereditary information. So that's why it's a messenger RNA, because it passes the genetic information from the DNA up to proteins. Yes, Dikshita is very right. Very good, Dikshita. Very good. Yes. Arundhati, you are also right. Very good. And tell me the function of RNA and tRNA also. The function of RNA and tRNA also. Yes, rRNA and our transfer RNA, which is tRNA. Yes, students, kindly reply. We have so many good students here. Please, all of you, I just appreciate if you reply me. I just appreciate it. Yes, Manpreet, you are very right. Very good. Yes, tRNA carries amino acids. Very good. Yes, Meera is very good. So tRNA is basically for the purpose of protein synthesis. So that's why tRNA is used. Yes, Jagriti, you are very right that rRNA is used basically for the, or you can say for the catalyzing the peptide bond formation. Yes, someone is very right that it encodes you can say the codon for the formation of proteins. So it has all the information along with it. Okay, good. Nitya is also here. Very good. Maya Kripa is hi. Hello. So Nitya is saying rRNA reads the DNA sequence. Yes, good. Yes. So tRNA acts as an adapter rRNA. Yes, very good. So you all have a good knowledge regarding it. So I am waiting for another students also. We have another students also. Ankita, Shivangi, Shraddha. Where are you all? Kindly reply, bache, please. So this is the only thing which we have to cover in our first lecture series. with this we have concluded our first day, and now we have to move to another lecture which is on today, that is our hormones. But before hormones, I have a small, you can say, video on central dogma, so I will share with all of you, then we will move forward directly to the hormones. And in between this video, I just appreciate all of you, if the students who have not given the answer yet, that kindly write their answer. I will check right after the video. Okay. Gunjan is saying transfer RNA is the key link between transcribing RNA. Yes, very good, Gunjan. So it's a key link. So if you students, tRNA is basically for transcribing the RNA and translating the RNA into the proteins. For that we have to need the specifically tRNA, which will carry the, you can say, the RNAs or messenger RNAs along with it. So just wait for 1 minute. I want to show you the video. I think this is the video maybe. Because some students are also new, and yes. that's all for central dogma. Inside it's looking like the helicopter. So I will share this video with you. Kindly see yourself, okay? I hope you will understand. This is only a video or animated video, so I will share it with you, okay? So that's all with our basically the thing which is our central dogma. One minute. Yes, this is much easy and also small here.
Cells. The most important task for a cell is to synthesize proteins based on the codes in DNA. But how are these codes read? What's the process that helps decode this language? And are proteins synthesized merely by the sequence of A, T, G, and C? The answer to all questions lies in the central dogma of life. But what exactly is the central dogma? Well, DNA makes copies of itself by the process of replication. Post-replication in every cell, the code of the DNA is cracked and transcribed into a messenger RNA. This mRNA is then read and the code is translated into proteins. This interesting concept is nothing but the central dogma. We know that the replication process is semi-conservative in nature. So now our focus will be on the two processes, namely transcription and translation. Let's begin with transcription first. As we can see here, the process in which mRNA is synthesized based on the parent DNA template is called transcription. The part or segment of DNA which encodes for a particular protein is called gene. So how are proteins made from the sequence of base pairs in the gene? Let's go in the reverse direction now. Proteins are a specific sequence of amino acids. So the various amino acids come together in a specific series to form a chain that makes up the proteins. Now to get the amino acids in a series, we need a set of instructions. These are present in the form of mRNA. And where does the mRNA come from? It is synthesized from a special template. Can you guess what this template is? Give it a shot. That's right, the template is a gene. So a segment of the DNA that encodes for protein is copied first. The copy is in the form of mRNA. This process of making mRNA is called transcription. Now can you tell me why this process is needed? Well, it's quite simple. If there is no transcription, then how will mRNA be synthesized? And if that's not done, then how will the proteins be made? Hence, transcription is vital. But it's not as easy as it seems. There are several enzymes and sub-steps involved in the process. We are referring to the steps like opening of the DNA double helix at a specific point, assembling the set of enzymes, gathering of nucleotides, and so on. After all these steps, an mRNA is formed. The mRNA is like an exact copy of the DNA sequence that codes the protein. The only difference between the two is that mRNA is single-stranded and the base thymine is replaced by uracil. Now that the mRNA is ready, it's all set to enter the cytoplasm. That is where the next step begins. It's the translation process. Let's have a look at this interesting process in the upcoming part. Imagine you're appearing for an exam, but the paper presented to you is in a language that you don't understand. You are prepared for the exam, but interpretation of the questions is a problem. What can be done? Some help from a translator would work wonders, right? So translation is the simplest way to solve this problem. Clearly, a wise move would be to decode the unrecognizable language and then attempt the exam. Now imagine this cell to be in a similar situation. Let's assume that this cell encounters a set of instructions for performing a task. But the instructions are written in a coded language. So all it needs to do is to translate this language and then perform the task. Any idea which process are we talking about? That's right, we are talking about the translation process. In continuation with the process of transcription from the previous part, let's now understand the next step of the central dogma of life. As the name suggests, translation means converting something into a simpler, understandable form. And what's getting translated here? That's correct, the code hidden in the mRNA molecule. The process of transcription helps in copying the code written on the DNA. That means it forms the mRNA. Now the mRNA comes out of the nucleus through the nuclear pore and enters the cytoplasm. Here, the process of translation begins. To begin with, let's find out the directionality of this mRNA molecule. This is the 5' end of the mRNA and this is the 3' end. And how do we know this? Are we randomly stating this? No, it's not random. We can recognize these ends with the help of specific markers. The 3' end has a continuous series of the A nucleotides. This region is called the poly-A tail. The 5' end, on the other hand, has a gap of methyl group. Thus, this end is said to be methylated. But why is the directionality needed? Well, it's extremely important. The assembly that carries out transcription binds to the mRNA near the 5' end. So how does the assembly know that this is the 5' end? The easiest way to recognize the 5' end is by the presence of this methyl gap. This is how the assembly understands to bind near the 5' end and not to the poly-A tail. Now what exactly is this assembly that we're referring to? Well, that's the ribosome unit. 2 units of ribosomes, one small and one large, form an assembly that helps in the process. Ribosomes are basically a complex assembly which acts like a molecular machine for protein synthesis. Remember the small dots attached to the surface of the rough endoplasmic reticulum? We find them in the diagram of a typical cell. These are nothing but ribosomes. So the ribosomes are usually made up of 2 subunits, a smaller and a larger one. The smaller subunit binds to the mRNA at the initiation site. The larger subunit binds and completes the assembly. Now the other type of RNA, called tRNA, steps in to get the remaining work done. At the end, a chain of amino acids is formed and released from the ribosomal unit. So doesn't this chain of amino acids look familiar? You got that right. It's our protein. The chain is further modified to achieve a proper configuration so that the protein becomes functional. This is how the translation process works. The code of mRNA is translated and the protein chain is synthesized out of it. The process of transcription and translation together are called as gene expression. The code of a gene is transcribed first and then translated into the form of proteins. There is an extremely important thing to note here. Whatever we've talked so far is all about the processes in eukaryotes. Such high level of organization is found only in eukaryotes. But we know what eukaryotes are, right? A cell with a well-defined nucleus and a highly organized system is a eukaryotic cell. On the other hand, cells which lack a proper membrane-bound nucleus and organization are called prokaryotic cells. Since they lack the proper membrane-bound organelles and nucleus, they do not undergo such complex and sophisticated processes. So the replication process in prokaryotes is slightly different. This was a brief idea about the transcription and translation process as seen in the eukaryotes. For more interesting concepts in genetics, tune into our channel.
So that's all with our Unit 1 or first day lecture series. I hope you are able to understand the functioning of central dogma, or how the functioning of transcription and translation has been done with this video very clearly, because it's a very good animation by which we are able to clear our all concepts. So are you able to get it all? Kindly reply please. So now with this our first, okay good, Kritika is okay good, and I also share the videos with all of you. I hope all the concepts regarding our first unit, which is DNA, RNA, we have nucleic acids, we have transcription, we have translation, and RNA types, also lots of things are there which have to be covered, because it's the first day on that one. So we have cleared it very briefly and thoroughly. So now we have to move forward to our next lecture, which is on hormones. So is everything is clear? Okay, everything is clear. Very good, very good. So good. Now we will move forward to our next topic, which is hormones. So have you known something about hormones? If you know, kindly let me know. So basically, what are hormones? We have so many students here. We have Anushri, we have Shweta, Rituja, Vishali. Kindly let me know. Or you need a break? Or is it okay? Shall we continue, or you need a break of 2 minutes or 3 minutes? Let me know please. Because hormones is a very important topic which we have to cover very briefly. Yes, very good. We have growth factors and practicing chemical messengers which carry the message from one location to another within the bloodstream. Very good. Okay, Omkar is also here. Okay, good Omkar. Okay, so good. So now we will, yes, Kumar is saying hormones are chemicals released in our bodies in trace amount which helps in regulation of body. Yes, it's very good. It's very true, Komal. Very good. Yes, chemical messengers, signals, secretly directed into our bodies or in the bloodstream. So okay, now we will move up to our next topic, which is hormones. We will take the break after hormones. So just let's start the topic first. Okay. So keep replying me also. Okay, the things which I am asking you, just give me answers also. We have so many students are here, so kindly reply. So okay, now we will move further to our next topic, which is hormones. We will start with protein hormones first. Yes. If you are seeing your syllabus, then in your syllabus we have to do protein hormones, steroid hormones, then we have a thyroid hormones. But before it, we must have some knowledge about what is hormones, what are hormones, what are their functions, and what's their need in our body. So we will start a little bit about hormones, and then we will keep up to the protein hormones. Okay. Yes, Vishali is very right that it is gets secreted in our blood. Very good Vishali. Yes, Geetika is also right. Okay, good. So keep replying. I will come back and then I will check your messages. Okay. So I am now starting with our hormones. So basically the science or the study of studying the hormones is basically endocrinology. Basically it's endocrinology. So we have different glands present in our body Like we have pineal gland, we have pituitary gland, we have parathyroid, thyroid gland is there, then we have thymus, then we have pancreas, adrenal gland. These are the glands basically. I'm just telling you about hormones basic. Then we have ovaries and testes. Ovaries in case of females and the testes in case of males. So these are the actually the, you can say, steroid hormones, the ovaries and the testes, and the others are the important hormones in our body. So we have pineal, pituitary, thyroid, parathyroid, thymus, adrenal, pancreas, ovaries and testes. But our main focus is on this sex hormones, or you can say steroid hormones, and the protein hormones. So basically it's in 1902 when we are able to we are able to come to know that hormones is there or something like is there. In 1902 we are able to know this thing. So it's, the theory is known as the hormone theory. And let's see what the hormone basically is. So substance released by one cell. So this is a one cell with nucleus inside. So substance released by one cell to regulate another cell. So this cell is releasing something to regulate the functioning of another one, synonymous with chemical messenger delivered through endocrine, neuroendocrine, neurocrine, paracrine and autocrine systems. Basically the substance is released, the chemical things is released to regulate the functioning of the nearby cells. So hormones can be lipids, like we have steroids, we have these prostaglandins, then we have the hormones which are proteins in nature. Basically either they are lipid in nature or they may be proteinaceous in nature. Like we have follicle stimulating hormone, thyroid stimulating hormone, growth hormone. All these hormones are protein in nature. So we have the types of hormones. We have lipid hormones, we have protein hormones, we have peptide hormones, and we have amino acids. But we have to discuss these two in detail, these two, lipids as well as proteins. Okay. So why the studying the hormones or endocrine system is important? Because maintenance of the internal environment in the blood, in the body. So the internal environment, to maintain, to regulate the activity of every cell, there is a need to send specific signals or specific chemicals. So that is why endocrine system is very important. Then integration and regulation of growth and development. That's how we grow, or how there is a growth in our body. It must be regulated. It's not like that when we sleep one day and suddenly in the next day we become 4 centimeter tall. It's not like that. So everything takes time. So it is regulated with the help of these endocrine hormones or endocrine glands. Then we have control, maintenance and instigation of sexual reproduction, including gametogenesis, even the development of the child. All these things, that how pregnancy happens, or how there are the hormones which are controlling the, you can say, all the things in the mother. So all these things are generally done with the help of these hormones. So these system or these hormones also helps to maintain homeostasis by integration and control, secretion of chemical signals. So this is a very simple, you can say, definition. The secretion of chemical signals is called hormones. And what are the hormones? Basically they travel through the bloodstream to act on the target cell. So suppose this is the intestine, or you can say any part of the body. So they travel within the blood. Why they travel within the blood? To reach on the target cell, where we have to send that particular signal or send that particular message. So they have to move within the bloodstream to reach the, suppose the if you are you must know it that a hormone or the gland is present in brain, and if something happens on our hand, we are able to know it. So the signals are transferred within the bloodstream. So this is the endocrine system. So we have endocrine system and we have nervous system. If you are able to know that we have nervous system also in our body, but nervous system performs short-term crisis management. The nervous system performs short-term crisis management. What does it mean? Suppose I am just there's a hot water. I just put my hand, then I suddenly take my hand back. Why? Because the brain gives me the signal or gives the signal to my hand that get back. Why? Because it is a hot water and it is dangerous to you. So it's a short term management. The nervous signals are very short term, they are very instant, that's on a spot. But in case of endocrine system, they are long term ongoing metabolic. So like nowadays, if you are aware of this thing, that lots of diseases are there which is because of your mental stress, as we can say, or some people say I did depression or some like this thing. So all these things are basically on the basis of hormones. Like sometimes we are very sad because the hormones of the sadness are more prominent, and sometimes we are very happy because we have the good hormones inside us. So we have stress hormones along with us, we have good hormones. It is up to us how to use these hormones, or if we keep thinking on one point or only on one topic, so we will just get stressed. That is the thing. So basically all the activity of our body, like suppose I am happy today because of the hormones inside, or suppose I am angry today, I am very angry because of the internal, you can say, the prominence of any hormone. So they play a very important role in the long term ongoing metabolic roles. So you have to know this thing. So how they transmit the signal, the types of cell to cell signaling, that how they transmit the signals. So basically what happens, the classic endocrine hormones travel by bloodstream to the target cells. We already discussed it, that from the bloodstream they will reach the target cell. Like this is a cell and they release a hormone. Suppose hormone A. Hormone A is released, and now within the bloodstream cell B will receive this hormone. So we have two types of functions, like they are showing actually two types of activities. One is by endocrine and one is by neurocrine. So cell A releases the hormone which by travel reaches the cell B. And in case of your nervous system, we have axons, or you can say neurons. So these neurons pass the information in the blood and it will reach to the second cell. So we have two types of endocrine, neurocrine, then we have paracrine also, and we have autocrine. In autocrine we are sending the signal to our own cell. So we have different types of hormones. So neurohormones are released via synapses and travel by the bloodstream. We have to focus on endocrine only, in which the hormones are transmitted by the bloodstream. And paracrine hormones act on the adjacent cells, the very nearby cells. And what are the autocrine hormones? Which send signal to own cell. They are just released and they are just providing signal to own cells. So we have four types of cells, or you can say four types of hormones. We have endocrine hormones, in which the information is passed through the blood from one cell to another. Then we have neurohormones, in which the information is passed through the blood but with the help of synapses. Then we have paracrine hormones. Para means nearby, and then we have autocrine hormones. Okay. So these are saying that why we are focusing more on endocrine. Why we are focusing more on endocrine? Because the hormone is distributed in blood and it binds to distant target cells. So with the help of endocrine, we are able to transmit the signal very far away. But in case of paracrine, the signal is passed only to the nearby cell. Only to the nearby cell, which is only the nearby cell. And in autocrine, the signal is passed to our own cell. So that is why endocrine is much important as compared to paracrine and autocrine. So endocrine system, the body's second great controlling system which influences metabolic activities of cells by means of hormones. And what they are? A ductless glands composed of epithelial cells that releases secretions directly into the fluid. So we have exocrine glands also, and we have endocrine glands also, but our main focus is on endocrine glands. Why we are focusing on endocrine? Because they are able to transmit signal or transmit any message to the long distance. So we have this thyroid, parathyroid, we have different types of hormones. So these are the broad, you can say, representation or the diagrammatic vision of the endocrinology, but our main focus is on these hormones: androgen, testosterone, estrogens, as they are steroid hormones. And then our main focus is on protein hormones, which is these thyroid gland. So we will study these in detail. Okay, rest are also very important, but they are not in your syllabus. So we will mainly focus on thyroid, and we are focused on the steroid hormones, or you can say sex hormones. So yes, this is another thing. The types of hormones in our body: we have protein hormones, which we have to study. We have steroid hormones, and we have amine hormones. Now what are proteins and polypeptides? Hormones from anterior and posterior pituitary, pancreas, and parathyroid glands. So the hormones which are basically, I will show you here. From here, from the brain area, that is from the parathyroid, or you can say from the hypothalamus and from the pituitary gland, they are basically the protein hormones, and they are very much important in our body because they are released from our brain area. So these hormones that they are released from pituitary or from hypothalamus, they are very important. These hormones are stored in secretory vesicles until needed. So what happened here? Basically, like this is the structure of spores, this is the pituitary. So these hormones, they are present there in the form of these round structures. So they are generally secretory vesicles, and they are only released when there is emergency or when they are required. So these are the main hormones in our body, the proteins hormones. Then we have steroid hormones. They are released from adrenal cortex, ovaries, placenta, all the hormones which are related to gonads, or you can say sexual reproduction. Then we have amine hormones, in which we will study thyroid. Now these days thyroid is very prominent. Lots of females, or you can say girls, have a thyroid problem. So it's very important hormone. We will discuss it in detail. So this is again the broad classification of hormones, but we will not discuss it as much. And this is how the hormones are synthesized, and then they are released. So let me show you. This is a messenger RNA. Messenger RNA on the ribosomes of the ER binds amino acids in the peptide chain called pre-pro hormones. So we have, this is the messenger RNA, and this is a pre-pro hormone. That the pre-pro hormone has no information at that time. Then when they will meet, that mRNA and pre-pro hormone, they form pro hormone. So enzymes in the ER choke of the signal sequence, creating an inactive pro hormone. So this is basically inactive at that point. Now what happens? This pro hormone, they passes from the endoplasmic reticulum through Golgi apparatus. So this is the Golgi apparatus, if you are able to see, and this blue pink will comes inside the apparatus. And now what happened there in the Golgi apparatus? What will happen? Secretory vesicles containing enzymes and pro hormone bud off the Golgi. The enzymes choke the pro hormone into one or more active peptides. So what will happen here? They will pass through the secretory vesicles, and now this pro hormone comes here. Okay. So the secretory vesicle releases its contents by exocytosis into the extracellular space. The hormone moves into the circulation from transport to its target cell. So now from this Golgi secretory vesicles, now it will release out, and it will just enter into the bloodstream. So this is the bloodstream where it enters. So the protein or any type of hormone has to pass from the important organelles of the cell, that is from the endoplasmic reticulum where we have mRNA. Even we see it in the video that these dots are mRNA. So from mRNA they have to enter into the, that is from the reticulum they have to enter into the Golgi body. In the Golgi body we have secretory vesicles. From these secretory vesicles they just move into the, so let me check whether you getting my points or not. So is it okay? Are you comfortable? Are you able to understand? Kindly let me know please. The diagrams are not clearly visible. Yes, Sakshi, you are very right that rRNA associates with the proteins to form the ribosomes. Okay. Amani Singh, ma'am, why should hormone exist in the form of amino acids? Would not it be more energetically favorable if it exists in as a 3D protein structure? Yes, Aman, actually this is the part of the proteins which we will discuss, and I will tell you then that why these hormones, not, and they, of course these are types of hormones. We have different types of, you can say, protein hormones, and we have amine hormones here also there. With the continuity of the PPT you will able to understand this thing. So are you able to understand what we have discussed? This diagram is visible to you. How they transfer from the cell to the bloodstream. Is it okay to all of you? Are you getting it? Please kindly let me know if are you getting it. Because today I made the PPT on the white surface. Yes. Okay, good. Kindly reply, bache. Where all are you? Just give me some reply so that I will continue it further. Yes, Aman is here. Okay, Aman. Thank you. Arun is here. Arun. Okay, good. Shraddha is also here. We have just start with the hormones only here, and we have to complete our whole PPT. I hope you will get the answers of all your questions. Yes, can see properly. Okay. Yes, ma'am. Okay, good. So we have just done the types of hormones and how they are transferred into the bloodstream. Okay, we have Jagriti here, we have Rituja, Megha is here, Jeet is also here. Okay, good. So we have one student which is yes, Sanjukta, I will share the PPT with you. I will share the PPT with all of you. It's my request to you or suggestion to you that kindly go through the video or kindly go through this PPT. If there is still any doubt, we will definitely share it once again. So there is not any issue. So don't be sad, please. Please don't be sad if you are unable to don't be sad if you are not able to get the PPT or the diagram. I will share it with you and then you go through it again, and if you have any type of doubt we will share. Please don't be sad, Sanjukta, please. Okay? Yes, please don't be sad. Yes, Devashish, but your name is Arun Mukherjee here. Yes, so it's writing Arun Mukherjee. That is why I know Devashish, but I don't know who is Arun. That is why I am saying Arun is saying Arun Mukherjee. Okay, good. Thank you, Sanjukta. Thank you. I will share the PPT with you. So you have not to worry, please. So Devashish is also here. Devashish, your name is Arun also. Okay, it's your dad's profile. Okay, very good. Okay, good. So I will remember now that it's you. Okay, good. So shall we continue it? So I'm just coming back to know whether you are getting it or not getting it. Just we will do something more, then we will come back to this review part Just let me complete a little bit more about hormones today. Okay, okay, good, good, good. So this is the how our pre-pro hormone becomes the hormone and it enters into the bloodstream. So you have to remember the 4-5 steps which are there in this from pre-pro hormone to the blood vessel, the hormones which are released into the blood vessel. So now we have number 1 hormones which are peptide or the protein hormones. So what are basically these? In some cases the pro-hormone is secreted and converted in the extracellular fluid into the active hormone. An example is angiotensin is secreted by liver. Yes, this is very important thing. So there is a hormone which is angiotensin. It is secreted by the liver and converted into, so angiotensin is basically inactive form. Liver released it, okay, and then it enters, you can say it gets converted into active form by enzymes secreted by kidney and lung. So what I basically we are want to say that pro-hormone, so angiotensin is an inactive hormone, or you can say pro-hormone is secreted and converted in the extracellular fluid into the active hormone. So these are basically the protein hormones which are generally inactive in form. Like we have so many hormones in the digestive system, and the angiotensin is there, and another hormones are also there which are in the inactive form. They become active, like we have trypsin, chymotrypsinogen, chymotrypsinogen. So these hormones are generally in the inactive form. They become active only when they form the signal, or they have some message from someone else. So this is the peptide and protein hormones. Then we have amine hormones, like we have thyroid stimulating hormone, TSH. So they are generally amine in nature. So they are group of hormones that are derived from the amino acid tyrosine. So the thyroid hormones which we will discuss, they are generally formed from the tyrosine, either thyroxine or epinephrine. So epinephrine is also an exciting hormone. It will release immediately. So sometimes we get excited by some type of news, like we have to go to watch a movie or for a shopping, so we just get excited. So these are these types of hormones. Then we have thyroid hormone. I will not take it yet, but I have to discuss it in detail. So I will tell you later about it. Then we have steroid hormones. Yes, we have to discuss 3 types of hormones. We will discuss steroid hormones, we will discuss protein hormones, the protein hormones which are in the inactive form. By some stimulus or something like that, they become active. Then the third ones are the steroid hormones. So, and the TSH hormones, they are derived from the amino acid tryptophan. Okay. So steroid hormones, all steroid hormones are derived from cholesterol. Number 1 point. They are derived from cholesterol and differ only in the ring structure and side chains attached to it. So basically they are you can say derived from cholesterol and differ only in the ring structure and side chains attached to it. So all steroid hormones are lipid soluble. Like in the very first slide I tell you that they are lipid in function, or they are soluble in the lipids. So they are basically cholesterol, which is a lipid soluble hormone. And then the types of steroid hormones which we have to study glycocorticoids, which release cortisol. Now I think this is your homework, this will be your homework. You have to tell me in the next section, then we will meet on tomorrow on the same day, on the same timing. You have to tell me the functioning of cortisol. Not now, but in the tomorrow session, the functioning of cortisol. Okay, so please write it down. What's the functioning of cortisol? And then we have mineralocorticoids. So aldosterone being most prominent. So in this we have aldosterone. I will tell you this. Then we have androgens such as testosterone. We have female hormones which is estrogens and progesterone. So these all hormones we have to discuss in the steroids. So these are the steroid hormones. We have cortisol, we have aldosterone, we have testosterone, we have estradiol, and we have progesterone. So basically these one are very important: cortisol, aldosterone, testosterone, estradiol, and progesterone. So you have to tell me the function of cholesterol only. Okay. So kindly note it. So we will just start steroid hormones. So what the steroid hormones are basically, they are not packed but synthesized and immediately released. Like the protein hormones which we have discussed, that they are present in the form of vesicles, but they are not packed but synthesized and immediately released. Okay, or for they are synthesized and then they are released. Okay, so all are derived from the parent compound which is cholesterol. We have to remember it, the steroids are generally made from the cholesterol and they are lipid soluble. Enzymes which produce steroid hormones from cholesterol are located in the mitochondria and smooth endoplasmic reticulum. So they are telling us that these hormones which are found from cholesterol, they are found in mitochondria and endoplasmic reticulum, that's the cell organaries. And these steroids are lipid soluble. Number one, that they are freely permeable to membranes, so are not stored in cells. So they do not need to be stored in a cell, rather they are synthesized on spot. They are not water soluble. Because they are lipid soluble, they are not water soluble, so have to be carried in the blood complex to specific binding globulins. So in the blood they will be, you can say they will be moved forward with the help of specific proteins, or you can say specific binding globulins. So we have corticosteroid, in which we have to study cortisol. We have sex steroid globulins which has testosterone and estradiol. So these are, you can, as these are insoluble in water, they will move in the bloodstream with the help of some type of globulin molecules. So in some cases, a steroid is secreted by one cell and is converted to the active steroid by the target cell. An example is androgen, which is secreted by the gonad and converted into estrogen in the brain. So they are saying that the functioning of one cell is controlled by the another cell. Like brain cell is controlling the functioning of the gonad cells, which are a sex cells basically. So brain cell is controlling their functioning. So these are our major steroid hormones. We have testosterone, kindly remember it. We have estradiol, we have cortisol, we have aldosterone, and one we have progesterone. Yes, there it is. So we have progesterone also. Also I have, let me show you one thing also. Also I have one steroid hormone PPT, but before it we will show you human endocrine. Let me know. This is the mechanism of hormone action basically.
The endocrine system is a very important and very involved system within our bodies. It allows our body to communicate over long distances and is a major player in regulating a stable, very important and very involved system within our bodies. It allows our body to communicate over long distances and is a major player in regulating a stable environment, or in other words, preserving homeostasis. This system employs the use of hormones, which are chemicals secreted from glands and enter the bloodstream where they circulate until exerting an effect on a downstream target cell. We are going to be specifically talking about the two types of mechanisms in which hormones exert their effect on target cells. But to start off, here is a brief overview of what hormones are and how they work. There are three basic groups of hormones. 1. Amino acid derivatives. 2. Peptide hormones. And 3. Lipid derivatives, or steroid hormones. For amino acid derivatives, these are small molecules and they come from amino acids like tyrosine and tryptophan. One hormone derived from tryptophan is melatonin, a hormone involved in our sleep-wake cycles. You can get tryptophan from foods like turkey, chocolate, or milk. Peptide hormones are made up of a bunch of amino acids. Some examples would be thyroid-stimulating hormone, oxytocin, or prolactin. Lipid derivatives, or steroid hormones, are considered lipophilic, meaning they have an affinity for lipid structures, which we'll take a look at shortly. They circulate in the blood bound to carrier proteins, so they usually last in the circulation longer than other types of hormones. There are two main mechanisms for which a hormone can affect a target cell. The first is by a non-steroid action. This mechanism is employed by an amino acid hormone or a peptide hormone. These hormones cannot freely cross a membrane on a target cell, so it first acts by binding a cell surface receptor. There is then an intracellular signaling cascade that occurs in order for the desired effect to take place. There are proteins on the intracellular side of the target cell that are associated with the receptor. Most often, this involves what's called a G protein. This G protein is found next to an enzyme that converts ATP to a molecule called cyclic AMP, or CAMP. This is something that makes this mechanism unique. Cyclic AMP is called a second messenger because it signals a cascade of events that eventually change the enzymatic activity in a cell to cause the target cell response. Examples of hormones that use the cyclic AMP second messenger system include ACTH, calcitonin, epinephrine, glucagon, parathyroid hormone, and ADH. The second mechanism is the steroid action. Because lipid derivative or steroid hormones are lipophilic, they can freely move through the membrane and into the cell. From there, they can either bind to a receptor within the cell, or they can move freely into the nucleus, where the hormone, or hormone complex, causes a change in gene activity. This then increases transcription and mRNA production, which then leads to an increase in protein production within the cell. Examples of hormones that exert effects via steroid hormone mechanisms are testosterone, estrogen, progesterone, aldosterone, and calcitriol.
Now, let's take a particular example, like from one of these steroids, and try to understand its mode of action at the cellular and molecular level. But if you don't know that how these steroids are synthesized inside our body, then you should watch my other video, which is in the playlist, and the link is given at the end of this video. So, let's talk about puberty. In case of females, the breast size increased after the puberty, and it is due to the effect of estrogen. Now, let's see at molecular level how this physiological phenomena, that means breast size increase, is triggered by estrogen. So inside the breast, just to give you a sense of the anatomy, there are several lobules, and there are milk ducts, there are majorly adipose tissues. These things increase in size as a female is crossing her puberty and going to adolescence. Now let's talk about how estrogen can work at a cellular level. So let's talk about one breast cell, a cell of a epithelial cell of the lobule, and let's see how its size would be modulated with the help of estrogen. So estrogen, like many other steroid hormones, work with the help of intracellular receptors. So what are intracellular receptors? As the name suggests, these are present in the internal side of the cell. They are not displayed on the membrane surface. Now, they could be present in cytoplasm, and most of the steroid hormone receptors are present in the cytoplasm, and on demand, when the ligand is present, they move to the nucleus. Now let's look at the structure of the steroid hormone receptor. So it has a ligand binding domain which binds to the steroid hormone. It has the DNA binding domain which bind to the response element, and it has some activation domain known as AF1 and AF2. So from the gene, when it is transcribed, its roughly protein structure look like this, and in the DNA binding domain there are defined motifs which help to bind the DNA in a sequence specific manner, and mostly these motifs are zinc finger motifs. We would look at it in details. Okay, so let's look at the structure again. So here you have quite a lot of domains, and this is a segment of the DNA which shows this kind of organization. So the transcription activation function 1 and transcription activation function 2 domains help in transcriptional activity, while ligand binding activation.
So I will share this whole mechanism. Okay. I could come to know that there is a buffering, and we have not done the mechanism part right now, so we will discuss about it later on. So are you able to get my voice now or not? Okay, is it fine now? The signals are okay from my side. I hope you will just wait for. Is it fine now? Can you let me know? No. Okay, I just switch off this video. I just switch off the video, maybe of some issues, and even we have to discuss the mechanism later on. Then we will go through this video. Okay, so need not to worry. So yes, it's still buffering. Yes, I am stuck here. But voice is there, but no video. Okay. I just got freeze. I don't know why. Maybe because we are just doing this. Let me close all these videos. Okay, now it is okay. Yes, now I think that's okay now. Maybe yes. Now the connections are okay. They're showing excellent connection. It's still buffering. Now fine. Okay, thank God it's fine now. Okay. So that's the only steroid hormones which we have to do today. I think it may be okay. Now it's clear. Okay. Maybe because there are 2-3 videos open along with the live stream, that is why there is buffering or some type of issues. But I just switch off all the videos because we have to discuss it tomorrow. Okay. Good. So just today we have done just basically the hormones, the type of hormones, how they are come into the blood and what are their functionings. So Gitika is saying it's better now. Okay, thank you Gitika. So that's all the hormone part basically which we have. Now lots of students are there who put lots of messages regarding the review paper. So if you are here, then kindly let me know what basically you want to know, because sometimes I am unable to give the proper reply to all of you. Also, I tried my best, but yet, if you have some queries regarding the review paper and how long it takes and where is, have you decided your topics or not? First of all, kindly let me know. Have you decided your topics or not? Nitya is saying it's good now, ma'am. Yes, thank you, Nitya. And thank God it's okay now, because it's very difficult if it's so much of buffering. Yes, Aman is also here. Aman saying it's better now. Okay, good. So kindly let me know, have you decided your topics or not? Because you are asking so many questions in WhatsApp group. So please be there and let me know what are the basically the queries you have, so that we will discuss it. Okay. So please kindly let me know that what do you want to know. Have you decided your topic or not? First of all, let me know it. Yes, please tell me, have you decided? The videos which I have shared, yes, definitely I will share the link with you also, so that you will be able to get the points. Today I have not downloaded the videos, I just take it from the YouTube, so I will share the link with all of you. Okay, not yet, but finalized few. Yes, please, we have two days already we have passed, so just be with your topic now. Even there are so many good students who message me, they have their topics ready, they even have their literature ready, so that's very good. So I also want you to just select the topic. So many students are there in touch with me, they have decided their topic along with the literature they have searched. Yes, the topic is, or yes ma'am, you are audible. Okay Meera, thank you. Decided the topic, wanted your approval once. Okay Renu, okay, definitely. If you have made the abstract, then kindly send it on my mail, so that I will check it and I will confirm it to you. Okay Renu? So if title is there, title on along with the abstract, kindly send it to me. Can you just explain the whole? Jinal, I will explain, but let me read some questions first, then I will explain definitely. Yes ma'am, topic is decided, Aman. Okay, good. Meera, but between the video voice was not clear. Okay Meera. Komal has decided the topic. Very good. And Shraddha is interested in cancer cell research. Yes, very good, Shraddha. You want to write the topic or a review paper on a cancer cell. Okay, good. Just share your views with me, please. Sunjukta, don't send this sad smileys. You can take 2 weeks, no issues, but be happy. I just want the the smiling smileys. So don't be sad, dear. If you want more time, just take it, but don't be sad, please. Yes. So Chandni has also decided. Okay, Chandni, all the students who decided, kindly let me know. So you have to write the title along with your name and your institution name, your email ID, and then the abstract. Okay. So you have to send this on a Word file to me. Okay, Aman saying they need more time. Okay, take the time. Time is not an issue, but at least start the review paper, because it will take lots of time. When you will go through it, you will see it takes so much of time. So Gina is saying, sure ma'am. Okay. So Sanjukta, be happy, don't be sad, please. These all are our hormones. If we will become sad, then we will become more sad and sad. Not sad, or you can say tired basically, because of your daily activity. Arundhati said topic is decided but need to go through some research paper. Okay dear, okay. just go through some research papers so that we will make the best. Komal is saying, what is the deadline for submitting the review paper? Actually, I give you the 10 days, that is from 18th or 19th to 29th. But if you are little bit busy in your own schedules, in your own classes, then most probably 30th of October. So within 30th of October or 1st of November you have to submit it. Okay, so do not extend it, then you can say extend far. You have to submit it in the last days of October basically. Yes, Sanjukta is now happy. Okay, good Sanjukta. Yes. So be happy always. Please be happy. Sandhya wants to write something, but I think she has not written something. So we have another students also. Where they are? Please. Now we are discussing especially about reviews. So please, Manpreet, please, Gitika, please, we all. This Aman is here, Jeet is here, Rituja is here. So please, and also please like my videos. Why you are not liking the videos? It takes lots of efforts to put in front of you the things in a good manner. So it will be nice of you all if you like the video, and also if you subscribe the channel. So just give me some support also, okay? Because these types of things give us motivation basically. So please be with me and let me know what are your queries basically, please. And I want to thank all of you that you are continuously here for such a long hours. So to remain there continuously is a very big achievement. So I think it's a big achievement for all of you, and I want to thank you all. Yes. I know you are just doing it for the first time, so definitely you will take some. It needs some time. Time is not an issue basically, but because now I am for 10 days along with you, so I have a proper time to talk with you, to guide you. But from 29th I have my bioinformatics class, so that's why maybe I will not get that much of time at that time. So that's why I am saying for these 10 days make proper use of me. So if you have any query you can ask me, okay? So that I am here only for solving your problem. So just share the things which you have so that we will discuss it better. So yes, thank you for supporting, for liking the videos, and also subscribe it, okay, my channel which is Crash Courses. And also just put your point of view also, please. Even you can message me also, but if I am unable to reply you or I will take time, then please understand this thing. So just now let me know what are your other queries. You just want time, I give you the time, and any other thing, your topic is ready. Like today is Wednesday, okay, today is Wednesday, so you have Thursday, Friday, Saturday. On Sunday, I want that you within these 3 or 4 days please send your abstract to me. Are you getting my point? Please send your abstract along with your names and your institute name to me. You have 3-4 times at least for abstract, so that I will come to know whether you are going on the right direction or you have to do some types of changes. So that's why I am saying that please up to Sunday or on Sunday, we have 3-4 days from now onwards, kindly send me your abstract, okay? And even I am free on Sunday, so I will be able to check it thoroughly. Are you getting my point? Geetika is saying, ma'am, I have not applied for project along with the internship. Okay dear, okay. But if you are 3 or 4 friends, then kindly collaborate and make an abstract. Okay. So just send me the abstract by the Sunday or Saturday evening, so that on Sunday I am able to read your abstracts and I am able to tell you whether you are going in the right direction or you have to do little bit of changes or modifications. Okay. So by this you will also get motivated. Yes, we have to read up to on Saturday or up to on Sunday. Okay. Ma'am, you will be actually Arundhati, I am not aware about this 25th ambiguity, because I have endocrinology section which I have to take it. So Jeet, why you are saying no? You are not getting my point. What are you asking? Kindly elaborate. So actually I have my bioinformatics section from 29th most probably. So I am not aware of it yet, but I will let you know definitely. Jeet, what are you saying? Acha, he is saying, ma'am will talk bioinformatics. Yes, definitely. So from 29th onwards, I have a bioinformatics session. That is why I am saying you that I am here for 10 days for 2 hours along with you and make proper use of me. You have any query, you want to know anything, you have any doubts, kindly let me know. If I am not able to reply, I will reply. Definitely I am replying to all of you. Sometimes we are busy maybe, so please wait and I will definitely reply. And even on this online platform I am here, you can ask me as many questions as you want. Okay, so there is not a type of hurry basically, but I want that at least prepare your abstract, at least select your title, so that it will be easy for all of us. Shraddha is saying, ma'am, please tell about how to write and publish the review paper. Shraddha, these are two different terms. First is how to write the paper. Okay, this is another thing, and how to publish it is another thing. So I will discuss here how to write it first. When you will write it, then I will tell you how to publish it. Okay, both are very different things. You have to write it first. When you will complete it, I will tell the whole class basically how to publish it. Okay, so don't worry, please don't worry, no issues. Someone is saying abstract on WhatsApp. No dear, actually on WhatsApp we are not able to make the corrections like that. So kindly mail me. I will share my mail. Let me write it. So kindly mail me, so that if there is some type of corrections I have to do, I will resend back to you. Okay, so no need to worry. Yes, this is my mail. Yes, Sakshi, I will tell you how to publish, but please don't be impatient. Just start writing, then I will tell you the whole procedure. Because writing is different thing, publishing is different thing. You will only publish when you have your paper in your hand. Then we will send it to the journal. Then journal will check the paper. Then journal will do lots of mistakes in the paper. Then they again resend it. Then we have to make the changes. Then we again resend them. So just be patient. Show some patience. I will tell you. Okay. Ma'am, please tell us how to publish. Sakshi is here. So yes, Shraddha, I am sending my mail. So kindly share your abstract, at least your topic, and how many students are there in a group for a particular paper. Kindly let me know, so that by Sunday onwards your title, your abstract is final, and you have to write the you can say the main part or the review part only. Okay, are you getting my point? Yes, yes. Don't worry. You don't have to take any type of tension. Just your first duty is to make abstract. That's all. Just make it. Then I will tell you the other things. Okay? Ma'am, will you help us in the plagiarism check? Yes, yes. I will help you. Just make the paper first. Then I will tell you the software, the online software for plagiarism check. Then after plagiarism checking, we will send it to the journals. Okay, Renu? So just even if your paper is prepared, then kindly send me or send me the title and abstract. And please mail me the abstracts, because how can I read the content on WhatsApp? Just mail me. So yes, yes, come on students, if you have some query, kindly let me know. But yes, don't be impatient, I am there along with you. Just write the paper. That's the only thing I want to know. I want to say. Write the paper, please. So if you have any other query, kindly let me know. Yes, one student is asking, Aman is here, that how that I think one student is there, how to start the review process. So I have told you before also that kindly select some topic, like you want to write it on breast cancer, or on cancer, or on molecular biology techniques. You then what you have to do? You have to do just one thing. Just select your topic and type it on a Google Scholar. Like the topic is breast cancer. So you have to write breast cancer review on Google Scholar, and you will find hundred of papers related to breast cancer. And then from those papers you have to select like 10 papers or 20 papers of your interest, and then from those papers you have to take the content, and then you have to add that content in your own paper, and you have to add the reference of specific paper which you have used. Like suppose I have used the paper of some scientist whose name is Roger. So Roger in 2019 asked, or 2019 published a paper. So I am taking their reference. So when I am taking any point from Roger's paper, I will add the reference of Roger in the backward, or in the on the last after the full stop, that this line is taken from the Roger. Are you getting my point? So if you are taking the work, or if you are using the work of someone else, you have to add the reference of that person. Are you getting my point? Otherwise it will come under plagiarism. What is plagiarism? That is you are copying the content of anyone else. But if you are adding their reference, that I am not copying the content, I just refer from that person, then it will not come under the plagiarism. So you have, if you are taking the content from some person, if you are taking, it's not only copy paste, no, you are just doing the reference purpose. So you will add her name or his name, and at that time you are just using this thing, or you can say the point, only for the reference purpose. I hope you are able to understand. So are you getting my point, Anushri? Shraddha, Renu, are you getting my point? Yes, if you are here, please reply me. Is it buffering now also? No, the signals are good. So kindly reply, please. Kindly reply me. Yes, some student had sent me the chat. I checked it. Not checked it, but I received it basically. So kindly reply me. Are you here? Then please share me whether you are getting the points. Now, where are the other students? Komal, Sanjukta, Arundhati, please reply, please. Jinal, are you getting my points? Sanjukta, Shraddha, Meera, Komal, please reply, please. So if you have any query, you can ask. You can please let me know. I don't know why the messaging are not coming. Kindly reply if you are here. Please reply. Yes. Okay. You understand. Okay, good. Where is Sanjukta? Oh, Sanjukta, you are here. Okay. Okay. Aman, where are you? Aman, Jinal, Komal, where are you? Komal, Gloris, Komal. So I think you are able to get my point. I think so. So that's all from my side today. Because I'm not getting any replies, I'm not able to know. So that's all from my side today. We will meet tomorrow on the same timing. Till then, take care of yourself and a very good night to all of you. We will meet tomorrow. Okay, I'm not getting the reply, so I don't know. Yes. Okay, then take care of yourself. Okay, bye to all of you. Bye. We will meet tomorrow. Good night to all of you. Good night. Please take care of yourself, please. Yes, bye.